thank you. I want to thank the organizers for giving me this great opportunity to share with you what my lab has been up to in the last few years. So the project I'm going to share with you started with a question that kind of follows me, or has been following me for the last kind of 10 years. What shapes how we perceive the world? And I think the talks we have heard this morning, people have mentioned that there are many things that kind of shape our perception. So if you have two people going to a concert or they hear a door bark, of course, kind of we're going to have different perceptions. For one of them, the sound could be pleasant and they're going to have like want to approach. And for the other, it could be kind of a scary sound and very loud and they want to retreat. And the question is what makes kind of this difference? And of course, things like genetic background or family history or personal history affect the way we perceive sounds or any sensory stimulus. And I think Ross mentioned also that things like task engagement, and many people in this room has shown that task engagement or attention or even locomotion affects the way the auditory cortex works and the way we perceive stimuli. So in the lab, we started thinking, do deeper internal dimensions like emotional states have the same influence on sound processing and perception? And when we think about it, emotional states are part of the driver of complex behaviors, right? If we feel joy or stress or anger for a long period of time, this will affect decision-making, learning, and memory. A little bit of stress could be good for you when you're studying for an exam, but too much stress could be detrimental. And when we think about it, sound processing or sensory processing and perception are the building blocks of these more kind of bigger and complex behaviors. So do emotional states actually interact with the way we perceive sensory inputs? And philosophers have been saying that the answer is yes for many centuries. So Kant said like all perception is colored by emotion, and there's this phrase looking at life through pink glasses, right? And I think intuitively we all think this is true. Like the doorbell could sound louder when we're stressed, and heels will seem kind of deeper and higher when we're despondent. And there are several imaging studies done in humans where they look at how emotional states affect memory and learning, and they saw changes in sensory areas. But they thought about it like a side effect of their kind of behavioral tasks, of their experiments, and they really didn't look much into that. So we want to ask, do emotional states actually modulate auditory processing and perception? And when we started this project a few years ago, we had to kind of choose an emotional state to start with, and we chose stress, chronic stress, because stress is kind of a big hammer to the whole system. It impacts body physiology, brain function, higher cognitive processes. So we thought, okay, this is a good place kind of to start this project. And also, we know a lot about the stress response. Like the HPA axis has been really well studied, so we know how it works. We know that at the end, we have cortisol and adrenaline are the two kind of main players that help our body get ready to the fight or flight. We also know a lot about kind of the different types of stress, right? Stress could be acute, could be chronic, could be psychological, it could be physical, could be something in between. It's kind of different if you're a man or a woman in a different genetic background. So we know a lot about the stress response, about the mediators, especially cortisol and adrenaline. So the question is, okay, so what do we know about how stress, chronic stress in particular, affects kind of sensory processing and perception in the auditory system? So what we know is that stress can influence the perception of stimulus that is already good or bad. So what I mean by this, if you have a sound or any type of stimulus that is bad, let's say after fear conditioning, so we know the chronic stress can affect the way we react. But most of the time, hopefully, we encounter neutral sounds, right? We hear someone talk, or we hear kind of drop of a glass or something like that, and it's not like an aversive reaction, hopefully. So the question is, what happens to all the neutral stimuli we encounter? So when the stressor is independent from the sensory input we're encountering. Let's say you're under chronic stress because of your job, your kids, life in general, and then you encounter kind of a sound. What happens to kind of the sound processing and perception then? So there are some hints that something is happening, but it's not very clear. For example, there's a paper showing that higher cortisol levels are indicative of a better performance in discrimination in humans. But then there's another paper showing that restraint stress could be actually bad for mice when they perform a similar task. So it's not really clear kind of what's going on. There's another paper showing that if you inject cortisol for 21 days into a mice or a rat, then you get a kind of a sound avoidance tag. So it's not really clear if at all and how does chronic stress affect sensory processing. What we do know from mostly kind of clinical reports that there's a link between kind of hypersensitivity in tinnitus, hyperacusis, and also in other disorders that are more kind of sensory in ASD with stress. The more stressed you are, the more your tinnitus kind of flares up. So we know that's their connection. So in the lab, we started looking, okay, how is the perception of neutral stimuli actually modulated by stress? And this is a project headed by Gatas, who is a PhD student in my lab, and Katya, she was a master's student and finished a few months ago. So what did they do to actually kind of try to tackle this question? We took the mice, we changed their state from normative to stressful, chronic stress, and then we looked at the auditory cortex with photon imaging and tried to see changes in sound processing, and in the end, we used behavior to test for changes in perception. So the first thing we kind of wanted to do was stress out our mice, and as you can imagine, it's not that tough. Mice are stressed very easily, so we decided to use restraint stress. So we put them in a tube 50 ml for 30 minutes. Restraint is a very, very common. It has been used for many decades. It works on mice, rats, monkeys, humans. Think about being like in an MRI machine for half an hour. That's more or less the feeling. Nobody likes it, but it's mild stress. It's not like a shock or something like that, but it's stressful. And then we want to actually measure, is the mouse actually under stress? So we use kind of different types of measurements. We use open field. We also use pupil, and we also use blood samples to measure corticosterone. And here I can show you kind of the results of the blood samples. So we can see that there is kind of an increase in corticosterone just after the mouse gets out of the tube, and it takes about 2 hours to go back to baseline. And we can repeat that every day to make sure there's no adaptation, and we see here we did for a week, 8 days, that every time we do it, the corticosterone levels go up. So, okay, we can stress the mouse, and now is the question, what's happening in the auditory cortex? So the experiment went like this. We did our virus injection, we injected GCAMP, and then we put the corneal window. After we have expression, we do a wide field imaging to kind of map the auditory cortex, and then we start imaging for a week of baseline. So every day kind of we image, we play in different sounds to the mouse, and a big advantage of the two-photon is that we can go back to the same cells day after day after day. So we follow the same cells through this baseline period and then through the stress period. So we're talking about the same cells, baseline and stress in the same animals. So we put the mouse in the tube for half an hour, take him out, and then do the imaging sessions. So this is the experiment. Okay, so what did we find? So here we have an example of one cell, 3 baseline sessions, and the same cell, 3 stress sessions. So when we started looking at the data, we saw that there seems to be kind of a reduction in the activity during the days of stress. Here we have it on a different sound level. You can see here also kind of the reduction. Here we have kind of a different mouse, a different cell, and we also saw it here. So we started thinking, okay, there seems to be something here. And when we looked at all the cells together, we have here the baseline activity normalized. And here we have the activity during the days of the chronic stress. So we clearly see a reduction in activity, especially in the mid-sound levels. And we can see here kind of the calculation, but we can really clearly see it here. So it seems that there's a reduction. This was in white noise, noise-induced activity, starting at mid-levels during chronic stress. So we said, okay, so we see this reduction in activity. This could be the result of changes in the baseline activity or changes actually in the post-sound activity. So what we decided to do is we divided kind of the data into pre-sound and post-sound. So for the pre-sound is the window of time before the sound was played. Post-sound is the windows after. And what we saw just right away is that we see kind of a shift on the whole data. It goes up during the stressful days. So we have this kind of 2 days of baseline, and then the first session, the third, and the fifth during the chronic stress. And we see a jump in the activity, meaning that the baseline activity is already shifting up as the stressor becomes chronic. And then we looked at the different sound levels, and what we saw is that as the sound is kind of louder, right, we expect to see a bigger difference between the pre-sound and post-sound, right? We expect a bigger sound evoked activity if we look at 55 as opposed to 45 dB. And this is what we see during baseline. But what was interesting that when we looked at the days of stress, especially the later days, this difference became smaller. So we found there was kind of a reduced difference between the pre-sound and post-sound activity during chronic stress, means we have a smaller sound-evoked activity or smaller response to sound when the animals are under chronic stress. And we can calculate kind of the relative change, which is the difference between the post and pre-sound activity divided by the whole kind of activity. And we can clearly see that we have a reduction in this kind of contrast between sound evoked, and it's not really spontaneous, but baseline activity during chronic stress. So we said, okay, this was all pyramidal cells, and we asked ourselves, okay, who's driving this process? So we went back to the stress literature, and we found that they talk a lot about changes in inhibitory and excitatory cells, both, and PV cell is kind of frequently mentioned. There are changes in the structure of the cells during chronic stress. So that's why we use a PV Crea-14 mice, so we could separate the activity of PV cells and putative pyramidal cells. So we were kind of excited that, okay, let's see kind of if the PV cells go up, this will explain why the activity in the pyramidal cells goes down. To our, I'm going to say disappointment, we found that the PV cells actually do the same as the pyramidal cells. We found that during stress, their sound-evoked activity is lower. I mean, it's not really surprising because I think in the last decade, we've seen many instances that PV cells act the same as the pyramidal cells, but it was a little bit disappointed. But we said, okay, so maybe the whole activity of the auditory cortex is going down because the brain wants to allocate resources to other sensory modalities, maybe to subcortical areas, so maybe everything is just going down and we have less responses to sound. Another option is, okay, maybe there's a different cell that is driving this process and it's kind of inhibiting both PV cells and pyramidal cells. So we went back to the literature, the stress literature, and we saw that there are also changes in SST cells during chronic stress. So we took a new group of mice. This time they were SST Cre. We injected Cre-dependent GCAM, and we looked at their activity. And to our pleasant surprise this time, we saw that they have an increase in sound-evoked activity. So as opposed to the PV cells and pyramidal cells that we see a reduction, here we see an increase in sound-evoked activity. Before I show you this graph for these plots for the pyramidal cells, this is how they look for the SST cells. So we can see that during baseline, they don't have much of a sound-evoked activity. The difference between the pre and the post is pretty small. But as the stress continues, we can see a separation of the signal and really see that there's an increase in the sound-evoked activity. So here we have the relative change for the pyramidal cells, and this is the relative change for the SST cells. And we'll clearly see that they go kind of the other way around. So we think that maybe changes in the activity of SST cells are driving the kind of reduced sound-evoked activity we found in PV cells and pyramidal cells during chronic stress. So here we have like all the cell types together, and we can see at the different sound levels how the activity of the PV cells and pyramidal cells goes down and SST goes up. And we think this could be serving as some type of kind of mechanisms to reduce the salience of auditory information being transmitted during chronic stress. After we found this, one of our bigger questions was, well, does this actually develop over time? We don't expect the first time we kind of encounter some stress in our lives to change our whole perception of the world, right? We expect this to be kind of developing when we're under chronic stress. So to answer this, what we did is we divided the stress sessions into kind of the first day the animal encountered stress. So we do the stress and then kind of the imaging. And this is for the different three types of cells. And we see some changes, but they're very minor on the first day. This is on the third day, and this is of the fifth day. And it looks like it is kind of developing and increasing as the stressor becomes chronic. We can kind of quantify this by taking the mean activity of the baseline and then the activity of the different days, so the first, third, and fifth day of stress, just kind of fit a line and take the slope, and we can clearly see that we have a reduction, kind of a negative slope for PV cells and pyramidal cells, but a positive slope for SST cells. So we really think that this is something that develops as the stressor becomes chronic, and it's not just like first day and kind of everything is changing. Kind of the next big question we had was, okay, so we saw this reduction in sound-evoked activity for pyramidal cells. Does it really matter? I mean, does it affect perception at all? So to answer that, we did a bunch of analysis. And our kind of main prediction was that we're going to see a reduction in loudness perception. In the interest of time, I'll just show you one analysis, which was to build a decoder based on our kind of neural activity. So here we have the sound that was actually played, and this is the sound kind of we predicted based on our neural data. We get kind of a nice prediction of the sound level. This is the baseline days. What happens during chronic stress? This kind of disappears. We don't have these kind of nice predictions anymore. So with this another analysis, we said, okay, so we think the loudness perception is going to change during chronic stress. And our next step was, okay, let's actually test kind of this hypothesis. To do that, we trained mice in a loudness perception task that was, I think, developed in the Polylab, which is very nice, where we train mice to lick to one side when they hear a soft sound, 40 or 45 dBs. If they do it correctly, they get a water reward. If they do it incorrectly, they get a timeout. On the other side, we teach them to lick in the other spout when they hear a loud sound, 75 or 80 dBs. Again, correct, you get a water reward. Incorrect, you get a timeout. What I really like about this task, that you have all the middle levels, right? Then we play 50, 55, 60, 65, and so on dBs. And here, we reward the mouse no matter what. So he can choose soft or loud, and he will always get a reward. That's why this is kind of the sweet spot to actually test its perception, because there's no right or wrong answer, just what the animal wants to choose. And this is how the task actually looks. The mouse has the two spouts, speaker, and can decide if to lick left or right. So what did we find? So here we have the lick loud probability on the side and the sound intensity. And because we trained mice on the 75-80, we expect this to be high, and we expect the 40-45 to be low, and we get really a nice curve where we can see that the mice are kind of on task because this is high and this is low, and then we get their choice in between, and we get kind of 50% of perceptual boundary, which is around 60 dBs. What happens during chronic stress? We saw that this curve shifts to the right, which means now that we get a perceptual boundary that is much higher, closer to 70 dBs, which means that a level like 60 dBs that was before considered, let's say, loud 50% of the time, now is considered soft most of the time. And to think of our kind of analogy to our real life, let's say someone is calling your name right in a room, they will have to speak louder or even shout to get your attention. And we think that's what's happening kind of during stress, that the focus of our system has shifted, and someone needs to kind of shout louder to get our attention. So after we found all these changes in sound processing and perception, our next question was how? How is this happening? How is kind of the auditory cortex being informed that we are under chronic stress, and how is it changing its activity? So now in the lab, we have 3 projects that kind of want to tackle this question. And the first one is to truly understand what's happening between the transition between acute and chronic stress, right? Because there are some studies that show different changes in acute. You will be surprised that we actually don't know that much about what's happening in acute stress in the auditory cortex, but there's one study showing increase in activity, for example. So what's going on in this transition between increased activity to decreased activity? How is it happening? So we have some preliminary data, and Lior, who is sitting here, she has a poster this afternoon, poster 43, so you're welcome to talk to her. I think we have some kind of really nice preliminary results showing that the effect of acute and chronic stress is really kind of different. Another project we're working on now is looking at the amygdala, cortical, and thalamic pathways during chronic stress to see if the amygdala, which is involved in chronic stress, is actually informing the auditory cortex and changing its activity to adapt to this new situation. And kind of the third project that I'm going to talk about for the last kind of minutes I have is what's the role of corticosteroid norepinephrine in modulating the activity of the auditory cortex. And I'm going to talk about norepinephrine. So norepinephrine under normal single conditions, let's say day-to-day, usually norepinephrine reduces spontaneous activity and increases sound-evoked activity. There's some discussions on exactly how this is happening, but I think most people agree that norepinephrine is actually improving the SNR. What happens during chronic stress? So under chronic stress, there's a dysregulation of the locus coeruleus norepinephrine kind of signaling pathway. And the question is, okay, so how does this affect the auditory cortex? We actually don't know. There's one paper that we could find that actually follow 21 days of restrained stress, right? They did stress for 21 days, took out the brain, and then look with HPLC the amount of norepinephrine per milligram in the outer cortex. And they found like a reduction. They found less norepinephrine in the outer cortex after 21 days of chronic stress. So we say, okay, this actually sounds good, because if we have less release or less norepinephrine during chronic stress, this would mean we'll have higher spontaneous activity and lower stimulus-driven activity. This is exactly what we found in our previous project. So we said, okay, does this really actually happen in vivo? So we decided to test it. So we used the gravity sensor, which is a sensor for norepinephrine. We injected it into the outer cortex, and we decided to image to see what happens kind of to the sound evoked activity. So just to test that it works, you can play, let's say, a loud sound, 90 dBs, and you really get kind of a jump in norepinephrine, which makes sense because that's kind of a scary sound for a mouse. And when you play something that's 30 dBs, that is kind of very, very soft, especially in the two-photon with the scanner, you get almost no change in norepinephrine. And we can see for different ROIs, that's a really big change. So we said, okay, so let's actually test what's happening during chronic stress. So we repeated the same protocol that we had before. We have a week of baseline imaging sessions, and then a week of stress sessions with the tube. So what did we find? So here we have an example of 1 day of baseline. So what we do, if we play a white noise at different sound levels, and we see the change in norepinephrine. So as expected, we see high changes in norepinephrine for 90 dBs, here in green, 75, a little bit lower, 60. And then for 30 dBs, we don't see kind of a change in the norepinephrine activity. So our kind of hypothesis based on the literature is that we're gonna see compromised norepinephrine release during repetitive stress. So we expect to see a reduction in the activity of norepinephrine when we play something that's very loud like 90 dB, and kind of no change for something that's low, because we shouldn't get a norepinephrine induced reaction during something that's not very scary or stressful. So here we have 4 different kind of baseline days, and we can see in all of them, we see a really nice kind of change in activity for 90 dBs, and then it goes lower. And I really like that you can see the different sound levels for all of them. Even if they look a little bit different, you see kind of the reduction for the sound levels in all of them. So what happened during stress? During stress, we really saw a reduction in the change in norepinephrine. So after the first day of stress, we can still see the difference here between 90 dBs, let's say, to 45 dBs. We see different, but as the days goes by and the stressor becomes chronic, we see that this kind of crumbles. Here is like less so, and here we almost can't see the difference between kind of the sound levels. So it seems that also here, as the stressor becomes chronic and develops, we see these changes in norepinephrine. And we can see the whole kind of all the ROIs here together, and we really see that repetitive stress actually attenuates any responses. And we think that this kind of matches this kind of one paper we found, that if we have an increase in spontaneous activity and a reduction in sound evoked activity, maybe these changes in norepinephrine are what kind of causing it, or are the mechanism by why the outer cortex is changing this way. So I hope I convince you that stress actually interacts with the way we perceive auditory stimulus. I showed that this actually happens for neutral stimulus as well, not only stimulus that have been conditioned. And we kind of think all the time why this is happening. And I think one of the reasons could be to allocate resources to other kind of areas of the brain, maybe subcortical areas. They're more responsive during chronic stress. And also it kind of reminds me of a lot of work we've seen, that locomotion of when we play a visual stimulus together with an auditory stimulus, we see this type of reduction in auditory evoked activity, and we find it also here. We see some of it being level specific, which was very surprising at the beginning. So we see it more in the intermediate levels, but not so much in the very high levels. So if we play 90 dBs, we're gonna get a response no matter what, even if the animal is under chronic stress. But this kind of also matches with the pain literature, because if you have a stimulus that is like so-so, then when animal is under chronic stress, or humans, they're gonna be more sensitive or less sensitive depending on the project to the stimulus. But if you punch someone, they're gonna react no matter what. So this is kind of similar. Stimulus are very, very loud, we're gonna respond. But when someone is intermediate, that's the place where stress has the most effect. We think that changes in SST cells are driving this process, but we still need to do perturbation experiments to see if this is really true. This develops over time, which kind of got our really kind of interest on what's the difference between acute and chronic. And as I mentioned, Lior is going to talk a little bit about that in her poster. And I didn't have time to show you, but we try different behaviors. So the loudness perception task I show you is what I think of a perceptual task, right? There's no right and wrong. You can't improve with training. If you think that the 55 is loud, that's fine. That's the kind of your perception, right? But there are other types of stats when you can improve. Let's say frequency discrimination. If we train, we can improve and we can discriminate between closer frequencies. And we found that those tasks are kind of less affected by stress, and tasks that are kind of more perception per se are kind of more affected. And we think that the changes in norepinephrine that I showed, this reduction in norepinephrine activity, really are driving kind of this process. We don't know if it's happening because there's kind of changes. We know there are changes in the locus coeruleus activity, and maybe that's why we see less norepinephrine in the auditory cortex, or maybe there's an increase in clearings in the auditory cortex itself. So we're thinking about different experiments to try to test that. So now we're working kind of with this, we have this working hypothesis that stress drives a kind of inverted U collapse of auditory gain via a neuroadrenergic decline. And now we have really different kind of projects in the lab trying to test different parts of this hypothesis to see if this is true. And with that, I want to thank my lab for all the hard work. I want to thank the funding sources and all of you for listening.
Try to get trainees' questions in there.
Oh God, this better be a good one then. Thank you, very nice talk. In the beginning you talked about the differential effect that is especially seen in SST cells, and then later you talked about a lot norepinephrine, so naturally for me the question arises, do we know something about the link between the two? And yeah, can you just tell us a bit more about what's known?
No, that's a great question. Yeah, so that's one of the things we're interested in now, looking at the different receptors in the different cells, like SST, PV, and also pyramidal cells, and to see if kind of there's a changes in expression of receptors, for example, for noradrenaline, norepinephrine, to see if that could affect more one cell than the other, or maybe they have an effect on both, and everything is kind of happening at the same time to really pinpoint the timeline and who's affecting who. Yeah.
Okay, thank you.
There's a mic behind you, yeah. Okay.
Thank you so much. That was a very interesting talk, and I made a lot of notes in it. So my thought is there's obviously somewhat a bidirectionality to sounds and their effect on our responses in terms of emotions. And so I'm thinking about misophonia, for instance. I don't know if mice have misophonia or not, but I'm just wondering, what are your thoughts in terms of, rather than changing just the sound level, but changing the salience of the stimulus? Because I thought it was interesting that you interpreted it that you might have a downstream reduction in the salience of a stimulus. But for instance, in misophonia with humans, they have an increased misophonic response when they're more stressed, and vice versa, the sound makes them more stressed.
Yeah, so I think the misophonia is not the same as stress. I think of a kind of a chronic stress especially as a state that we can kind of go in and out of, and it changes kind of, it modulates our sound processing and perception. I think the mechanism of misophonia might be a little bit different. It has kind of a longer term effect, has kind of the genetic background to it, as much as I know. So I think it's not the same mechanism, but I do agree that one informs the other, that kind of our change in perception then informs our changes to kind of auditory processing and so on. That's why we're also interested in the amygdala, to see how everything goes together, because what you kind of mentioned also about the connection between, let's say, emotions and sounds, everything goes together. So we think we're going to see kind of these changes between the amygdala, auditory cortex. We have some results from the IC as well, so we think it's also kind of involved, and at the end it's like all together making the changes in perception. Yeah.